Hello everyone, my name is Saeko Tahara. I'm a medical student at Tsukuba University in Japan. I will have a presentation about unmeasured ChIP-seq and its impact. The title is The Potential Impact of Unmeasured Transcriptional Factor ChIP-seq Data on Omics Studies. Recent attention on human genes is imbalanced and concentrated on several specific genes. Actually, a similar trend applies to TFs. Our preliminary survey finds that only 10% of TFs occupy half of TF-related publications. ChIP-seq can reveal the target regions and the target genes over TF and have been deposited in databases. The collection of ChIP-seq data have been utilized for enrichment analysis and prediction for regulatory factors. These facts prompt us to speculate that imbalanced research attention leading to imbalanced ChIP-seq data, resulting in impaired accuracy of the downstream analysis and inhibiting new findings of regulatory TFs. So here, we conducted a systematic investigation to determine the prevalence of unmeasured and measured ChIP-seq. For the systematic analysis, we used metadata in ChIP atlas and several databases, such as NOC-TF, TF marker DB, and literature data, Gene2PubMed. First, we identified unmeasured ChIP-seq based on TF expression. We defined unmeasured ChIP-seq by the existence of ChIP-seq samples and enough TF expression. In Figure 3, rows are TFs and columns are tissues, and this red color indicates measured ChIP-seq. We found that about 80% of TF and tissue pairs were not deposited in ChIP atlas. To identify unmeasured ChIP-seq, we checked the existence of ChIP-seq samples and TF expression. This blue color is unmeasured ChIP-seq. We found that about 25% of TF and tissue pairs were unmeasured, even though enough TF expression. Next, we identified and prioritized unmeasured but potentially important TFs and cell type classes. This importance was defined by the number of differentially expressed genes, DUGs, in TF knockout or knockdown experiments, and cell-specific TF markers. First, we plotted the number of DUGs in TF knockout or knockdown experiments and the number of publications. In this scatter plot, each dots are TF and cell type classes pair, and the red points are measured, and the blue points are unmeasured. We found that there was no significant difference between the number of DUGs in measured and unmeasured ChIP-seq, suggesting the existence of unmeasured but functionally important ChIP-seq samples. Additionally, we checked cell type specific TF markers and selected cell type specific TF markers and chose 8 candidates of unmeasured but potentially important ChIP-seq, as shown in Figure 7, blue ones. In summary, we identified unmeasured but potentially important ChIP-seq and selected 8 candidates. But what ChIP-seq should we do next? For answering the question, we are trying to check the unmeasured ChIP-seq importance in terms of regulome reconstruction, as shown in Figure 8. Moreover, what's the impact of unmeasured ChIP-seq on omics studies? The results of TF binding enrichment analysis and omics studies are dependent on these ChIP-seq data. So new findings are potentially inhibited by unmeasured ChIP-seq samples. This presentation was supported by JSTMRI program and ISCB and GLBIO through the Travel Fellowship Award. I'm so grateful to everyone who provided this opportunity. Thank you for listening.